Okay, I see it's recording now.
Yeah, that's good.
All right. So what I'm going to be talking about today, so I prepared a presentation. Um, I'm mostly going to be spending a lot of time talking about the science that's underlying coronavirus. So the basics of sort of the viral life cycle, how it's transmitted, how we test for it. I'll spend a little bit of time talking about medicine and then the public health aspects of this. And then there should be plenty of time for Q&A after. So if people have remaining questions. And really quickly, for those of you who don't know me, my name is Danielle. I graduated from Colonie in 2014, and I am a biologist. So my sort of main area of expertise is like cell and developmental biology and genetics, but I'm also broadly trained in general biology. And I just want to have this little disclaimer here that I am not a medical professional, so I'm not a doctor or a nurse. And so I'm here to provide you information, provide a scientist's perspective, but please don't construe anything I say as medical advice. Okay, so starting off with the science, what are coronaviruses? So coronaviruses are a family of viruses. They look like this. The reason why they're called coronaviruses is because the word corona means crown, and they have these spike proteins that give them the appearance of having a crown. So here's what they actually look like under an electron microscope, and here's a sort of diagram of what they look like. And so you can see that they have a sort of outer shell that is made up of proteins and lipids, just like our cell membranes do. And these lipids are also, known as fats. And this explains why washing with soap and water is really good at inactivating these viruses, because it makes this outer shell fall apart. Another thing I want to point out is that, like us, viruses have genomes, but unlike us, their genomes are made out of RNA. So RNA is a molecule very similar to DNA, and this will have a little bit of importance later. But just want to point out that their genome is made of RNA. So a lot of people have been wondering, have never heard of coronaviruses before. So the coronavirus you're hearing about in the news is not the only coronavirus out there, and it's not the only coronavirus that infects humans. So there are 4 coronaviruses that just regularly circulate through human populations. So there's a good chance you've had one of these before. If you've had a common cold, they're caused by many different types of viruses, some of which are these coronaviruses. So maybe between 1/5 to even 1/3 of common colds are caused by these coronaviruses. In the past about 20 years, we've seen the first emergence of very severe coronaviruses. So for a long time, people didn't pay much attention to the virology of this particular family because they only cause very mild disease. But in the early 2000s and the early 2010s, there was the emergence of SARS and MERS. So these stand for Southern Acute Respiratory Syndrome and Middle Eastern Acute Respiratory Syndrome. And these were very severe outbreaks, but they were very small in nature and were contained. SARS-CoV-2 is the scientific name for the virus that is causing COVID-19. And so I'm going to, throughout this presentation, be mostly referring to it as the novel coronavirus to avoid confusion with SARS, which is a different virus, but they're very closely related. And this point will come back later because this is actually helping us a little bit with being able to approach how to treat and how to understand this novel coronavirus. So SARS-CoV-2, you might see sometimes, that just means the novel coronavirus, and the disease it causes is COVID-19. So people have been asking, where did this come from? In the, about last month of last year, there was the emergence in the Wuhan in the Hubei province of China. Some patients were coming down with a new respiratory illness that had severe symptoms that didn't have a cause that was understood. And so the medical professionals who were treating these patients and the researchers that they were working with determined that it was caused by a coronavirus, but it was not caused by one of the 6 coronaviruses that to that point we knew existed in infected humans, which is why it was named initially the novel coronavirus. It caused SARS-like symptoms, so sudden acute respiratory syndrome, in the people who are hospitalized, so pneumonia-like symptoms, potentially leading to lung failure. And I want to point out that you've probably at least heard some people referring to this as the Chinese coronavirus, or other monikers like that. And I just want to really sort of say this very unequivocally. There's no good reason to say that. There's nothing special about the fact that it first emerged in China. It could have, if, we turned back time and replayed this, a pandemic could have appeared any place. And this affects everyone at with equal opportunities. So please do not be using this language that can fuel racism. And we've seen things like this happen before, like when HIV first emerged and people referred to it as a gay autoimmune disease at a time when that was considered very stigmatized. And this actually led to policy decisions that held back research and led to people dying. So this is both be a good person and also recognize that the language you use actually really matters. Okay, so where did this come from? How does a new virus emerge? So a new virus doesn't just pop out of nowhere. What happens is that there was an existing virus that was existing somewhere else that through random mutation gained the ability to now infect humans. It's known that bats are natural reservoirs for coronaviruses in the same way that we're natural reservoirs for influenza. So we have flus that, every year humans have flus. They're just part of our lives. They're going through us. Same with bats. They have all these coronaviruses. They're natural reservoirs for it. And in the same way that, for example, human diseases like leprosy have jumped to animals like armadillos, non-human animals can harbor viruses that can then potentially, through evolution by natural selection, acquire a mutation that confers the ability to infect a human. And it appears that this actually happened in 2 steps. So the virus jumped from a bat to something else, and then from something else to us. And we don't know what that is yet. You may have heard some speculation on what it can be, but we do not know yet what that intermediate reservoir is, and we may not ever know. And there was a research study that looked at the genetics of the novel coronavirus and compared it to the coronaviruses that exist in humans and bats and other known coronaviruses, because by doing these genetic analyses, we can make family trees and try to understand where these viruses come from. And I just want to point out that this really unequivocally showed that this dispelled any rumors, no matter how, some people were wondering, oh, there are research labs that work on coronavirus. What if this was a human engineered one? That is completely untrue. It does not have sequence similarity to any of the lab strains in a way that would be expected if it had originated in a lab. And there is no evidence of any genetic manipulation using human designed genetic tools. So this came from animals, and it's been around since late November or early December, with the disease first being recognized in late December. All right, I had some questions that people submitted about how is this different from comparable viruses. So I have this really nice infographic that was created by the scientists at BioRender and shared by Kiko Iwasaki at Yale University, who is a really great immunologist. And you don't need to know every single word on this diagram, but what I want to highlight is a few things. So this is based on a lot of the early statistics that are coming out of China, because the virus originated in China, that means that we started being able to gather data from its spread earlier than we have from other places where it popped up later. And it seems that, and I have these little tildes here because these are really rough estimates, because again, this is really, really new. So we haven't had the time to really get super great statistics on it, but it appears to be at least twice as contagious as the flu, meaning that it spreads very easily, with a much higher hospitalization rate, and it's much more deadly. I want to point out that when I say this, it's really important to be considering this from a statistical perspective. So for example, you see the case fatality rate here showing an estimate, and this might be off by a couple percents, but anywhere between 1% and 5% we've been, would be a sort of reasonable average. And that's a pretty small number for every individual person who gets it. If you get coronavirus, you maybe have a 1% chance of dying. That's still pretty high, but like not, a 1 in 100 chance is the odds are in your favor. Yeah, it's really important to consider that, like on a broad scale, if you have a 100-sided die that you roll, the chance of you rolling a 100 is low. But the more people it infects, the more likely that this low probability event will happen many times. And so it's the combination between its slightly high sort of danger, as well as the fact that it's a lot of people are getting it and there's no existing immunity in the population, because we have not had this type of coronavirus before, so nobody before this outbreak had antibodies against it. And something that I wanted to point out as well, and I'll get to a question in a moment, is that the reported death rates in China, again, where we have the best statistics so far, was much higher in the epicenter than in surrounding areas. And this is suggesting that a lot of the elevated fatality rate in the epicenter is likely due to differences in healthcare availability. So if you have one person who has the COVID-19 disease and a whole hospital to take care of them, their chances are pretty good. But if the hospital systems are overwhelmed, everyone's chances get worse because everyone gets a lower quality of care and there may not be enough resources. I have a question about the testing. I'm going to get to the testing in a little bit, so I will answer your question later in the presentation. And my cat is trying to get under the door. Okay, so people also asked, so they were interested in how is this different? In terms of just the situation that we're dealing with, now I'm sort of zooming out. There are some similarities, but it's really an apples to oranges comparison in many ways. The comparison is helpful in some ways, but it's not like this is just a worse version of the same thing we've seen before. It's a different version of something we've never seen before. So SARS and MERS, again, the most closely related human coronaviruses, had outbreaks in this century, and they were widely contained. So I believe that they both infected less than 20,000 people at least. And I'd say in terms of, somebody asked, like, have we ever had measures like this being occurring? And maybe the most similar would be the 1918 flu, where there were things like closing of bars and schools, and there were temporary hospitals built. But again, that's different in many ways as well. The flu has different, the virology of the flu is slightly different than the virology of the coronavirus. And then, of course, there are many people alive today who remember the sort of fear of the emergence of HIV. And so there are some comparisons to be made to that. But I want to point out that this is something that we're dealing with for the first time, and there are aspects of what we're going through that are very different than anything we've ever encountered. Okay, so how is the coronavirus transmitted from person to person? It appears that there's three major ways. So the first being droplets or aerosols, and I want to clarify what the difference between droplet aerosol transmission is and airborne transmission, because a lot of times in our regular usage, we use those words to mean the same things. So if you sneeze, if you've ever sneezed and your hand was wet after, also just sneeze into your arm, but you know what I mean. You know that you produce droplets, and that is the main way that the coronavirus is able to get from our respiratory tract out into the world. Aerosols, typically you can think of them as much smaller droplets. So like Febreze, if you spray it, it has droplets, but they're so small that they sort of float like a cloud. This seems to be possible, but we don't generate aerosols quite as often as we generate droplets. So often aerosols are being produced in hospital settings, like when a person is actually on ventilation, that will produce aerosols. Airborne is slightly different. That assumes that it can just float in the air. Droplets are slowly falling down and settling on surfaces. So it's not just hanging out in the air on its own. It's always associated with sort of a droplet. There's surface transmission. So again, if a droplet lands on a surface, it seems that they're at least semi-stable on different surfaces for different amounts of times. With, for example, like on cardboard, I think they can hang out for like 24 hours, about. This again is early studies, but if you touch something that has the coronavirus on it and get that on your hands, and then touch your face, that gets it really, really close to your airways and mouth and gives an opportunity for it to get in. And then there's some preliminary evidence, and also based on what we know about other coronaviruses, that fecal oral transmission can happen. So, and this is, it's pretty intuitive to most people that if you get poop droplets or little, things like that, that is a way that things can be spread. So just practice good hygiene. And I think that understanding how the virus is transmitted also helps us understand why we're getting the advice we're getting. So this is why washing hands is really important. I mentioned before that these viruses are encased by lipids, so the soap will break them up and they'll fall apart. Not touching your face, because that's how this can often be transmitted, and then keeping 6 feet away, because the droplets that we generally produce by a cough or a sneeze are often going to travel 6 feet or less. And so that's why these guidelines are being set up. Something else about the way that this is transmitted that I think is really, really important, and I'm going to remind you of this a few times throughout the presentation, is again, by looking at the data from the earliest spread that was happening in China, it showed that a lot of people who did not show symptoms, so they didn't even know they had the coronavirus, seem to be responsible for infecting other people. So a substantial number of the cases seem to be transmitted by people who don't even know they're sick. And this is one of the reasons why I mentioned that the virus is so infectious, because if you can compare this to something like Ebola, for example, where most people who have Ebola are not able to get up and walk to the grocery store and infect people because it's so severe in every person that they're not able to transmit it very well. But if you don't even know you have it, then you might go out into the world and spread it much more easily. And so this is contributing to a lot of the high infectiousness of this virus, as well as some of the reasons why we're having to deal with it in the ways that we're having to deal with it, because you can't just automatically tell if somebody is sick and infectious. So the next question is, how does the novel coronavirus infect human cells? And something I want to point out is, in this part of the presentation, I'm going to try to keep this at a very a simpler level. But if people have additional questions on more of the details, I'm happy to go into them in the Q&A session. So the coronavirus life cycle, this is taken from a review on coronaviruses in general, again, because we know a lot more about other coronaviruses than we do about the novel coronavirus. But coronaviruses bind to a protein on the surface of the host cells. These proteins have other purposes that, they are not designed to be a coronavirus receptor. They're being repurposed by the coronavirus as a receptor. The virus, it's like a bubble fusing to another bubble. It dumps in the genome, and then it uses some of the host machinery to both translate some of its proteins, and then a combination of the host machinery and the proteins that are being translated from the virus are used to replicate its genome, make more of the proteins that can comprise the virus, and then they are released through regular trafficking pathways in the same way that your cells are able to release certain contents through exocytosis. So I mentioned that the coronaviruses will use a protein as a receptor. Again, these proteins do not exist in our bodies for the purpose of being a viral receptor. And in the case of the novel coronavirus and the SARS-causing coronavirus, they use the same protein. And this is before, when I mentioned that the novel coronavirus and the SARS coronavirus, and some people are calling this the classic SARS, why it's important that they're closely related. It gave us a head start because we didn't have to start from scratch and try to figure out what protein in the cells of humans does it bind to? We checked if it was the same one as SARS, and it was. So it gave us a head start. The protein is called angiotensin converting enzyme 2. You don't really need to know about like the physiology of this to understand how the virus uses it, other than it has a normal role in your regular body physiology for maintaining proper blood pressure. But also by, it happens to bind really well to the spike protein of the coronavirus, and that's why it can be used as a receptor. So there's been some really heroic work that's happened already on this. So what I'm showing you here, and again, I'll point out the parts of this diagram that are important to understand, and you can ignore the rest. But what you have here in the sort of purple, blue, gray, and pink, this is the model representing the molecular structure of this angiotensin converting enzyme. And then what you see in this sort of gold color, they're just showing the portion of the viral spike protein that binds to it. And you can actually see, and if any of you have taken molecular bio with Mr. Goldberg, you may know a little bit more about this, but the amino acids on the surface have different charges and different sort of biochemical properties that if there's for example, a negative charge that matches up closely to a positive charge, those are going to like to be next to each other. And so there's about 6 residues here. So 6 different events of stabilizing this interaction. And that's why this binds really well to the angiotensin converting enzyme 2. Also, I want to point out that the ACE2 molecule that humans have is slightly different than the ACE2 molecule that other organisms have, and that's why the virus is specific to humans and then whatever host we got it from, and why it's unlikely to easily infect other things, for example, mice or dogs or cats. And someone said we did learn it in Goldberg's. Excellent. So next up, I want to talk about how do we test for the novel coronavirus. There's been a lot of talk about this, and so we're going to talk about the basics behind this for a moment. So again, I want to keep this down at the simplest level I can while still being accurate and providing the right information. So if you have more specific questions about this, happy to go into it later. But essentially what is used is an assay called qPCR, or quantitative polymerase chain reaction. You may have learned about just run-of-the-mill PCR in AP Bio or Molecular Bio or potentially even Regents Bio at this point. But what we do is, again, I mentioned that the coronaviruses have RNA as their genetic material. RNA can be copied into an exact DNA version of the RNA, so that's the first thing that we do. And then we can do PCR on it. And what PCR does is if there's a particular region, again, we know that DNA and RNA have very specific base pairs, or sometimes you call those the letters of the genetic code. And so you can think about the sequence as being very unique in certain places. And so what happens is we can put things called primers, which are single-stranded DNA molecules that can match up specifically by base pairing, because DNA can base pair one strand to the other, and they'll only be able to bind where there's an exact base pair match. And so what happens, so if there's the base pair match, what we do this chain reaction where every single time we do a cycle of this reaction, you double the copies of that particular sequence. So you can see over many rounds, you end up with a lot of the same sequence amplified. So if you don't have any place for these primers to bind, you won't get anything. But if you do, you'll get twice as many every round. And qPCR has an added component to it that essentially, I'm going to simplify this, but every time you have a successful round of replication, a fluorescent molecule is released and allowed to fluoresce, which means that if you measure how brightly the reaction is changing, then over time it becomes more and more fluorescent if you are getting amplification, which will only happen if the primers match a sequence. And so if the primers have nothing to match to, such as if there's no coronavirus RNA present, you don't get any new fluorescence. And so how do we interpret this? First, I want to talk about what the importance of controls and what this means. So when you do an experiment, you also have to do a control to be sure that you can trust your experimental methods. And so the way that I always like to teach, oh, got someone ringing our doorbell. The way that we teach controls is that you need to have positive and negative controls. And I think an intuitive way to understand this is if you're running a test, you need to make sure your test works and that it's not giving you false negatives or false positives. So a test that many people are familiar with are a pregnancy test. So if I give you a pregnancy test that's never been tested before and you say, okay, how do I know that this brand of pregnancy test works? Well, first, if you give it to a person who is definitely pregnant, then it should say that they're pregnant. And if it doesn't give you the result you expect, then there's a problem with the test. It's giving you a false negative. Likewise, if there's a person that you know is definitely not pregnant and you give them the test, so I picked Santa Claus. If you give this person a pregnancy test and it shows up as positive, then you know there's something wrong with the test. And so what we do is whenever we're doing a diagnostic test, we also do a positive and negative control so that we know we can trust the results it gives us. And if there's something wrong with the control, we don't have to trust the results it gives us. So the same is true with the novel coronavirus. So the CDC tests that were produced that you've probably heard about in the news came with positive controls. So this is RNA that does contain the sequence from the coronavirus genome that the PCR is trying to amplify. There's a negative control that should not have that sequence, and so you should not get amplification. And so again, when you're doing this assay on patient samples, you would also run the positive and negative control to make sure that you can trust that your reaction actually worked. So here's how you would interpret the results. I'm going to go through these one at a time. So this is actually the way that the original CDC test was set up. So to be double sure, they had two different versions of the PCR, one that recognized one sequence in the novel coronavirus, one that recognized a second sequence, just to be double certain. They wanted to make sure that they're crossing their T's and dotting their I's. So again, we know the positive control, you should get a result. Okay, that's good. We got it, we can trust that. The negative control, you shouldn't. And then the patient one we don't know about yet. They also used a third reaction where they did looked for a sequence that was found generally in all coronaviruses. So this would tell you if a person has a coronavirus, but these first two tell you if it's the novel coronavirus. And I have a question, what is the gene you're amplifying in this PCR? I'm not sure exactly which one was being amplified using the CDC test, but there are multiple different qPCR tests that are happening that have different targets. So when you hear about a new test was developed, and you're like, how did someone develop a new test in 2 weeks? It's because they just use different primers. So there are different versions of the test that target different parts. But one thing is, so the question that I have is referring to the S gene, which is the spike protein, which is the protein that the virus uses to bind to the receptor. And there's no reason it has to be from a particularly important part of the genome. It just needs to be a part of the genome that is specific to coronaviruses for it to be effective. So going to this table again, we see that if you have an infected patient that you run this assay on, the positive control should always come up positive. The negative control should always come up negative. Okay, that means it's working. And then the patient comes up as positive for all three of the replicates. So we would say, okay, this person has coronavirus. And it's the novel coronavirus. Inconclusive results happen if you get it coming positive on some and not the others. And so this we would run again because that suggests that there might be something wrong. And so this adds, by doing 3 different tests for the same person, it adds a level of stringency, because in theory, if they have it, it should always come up positive on all 3. So this is an extra safeguard against false positives and false negatives. Which brings me to the big question about why has testing been so delayed in the United States? There's certainly political aspects about this as well, about the choice to use exclusively tests derived in the United States rather than ones provided by the World Health Organization, but I'm going to focus specifically about the science behind this. So the CDC quickly developed a test, and again, developing a test is something that can happen quickly because we do qPCR all the time. It just means picking new primers so that they work for the novel coronavirus. But what happened was when the CDC sent their tests out to state labs, there was a problem with the controls. And this is why I've spent so much time talking about controls, because I want you to understand that concept. So what happens was people were getting the sequence that's supposed to have no coronavirus RNA in it. Some of the tests were coming up positive when they should have come up negative. And so this made the clinicians who were running these samples, red flag, say, I don't know if this test is working. And so everybody who had this problem had to halt their test usage and divert all their samples to the CDC instead of being able to test them. And this had to do, the reason why is a faulty reagent, and it hasn't been released exactly what about it is faulty. I have some ideas about what could have been wrong with it that could cause a false positive. And so somebody's asking me, so the rates are, about how this relates to the rates and the reliability about data. I would say that we know how many confirmed cases of coronavirus there are. And I think what you're trying to get at is that the infection rates in the United States, the confirmed number of coronavirus cases is going to be much lower than the actual number because we're not testing everyone. So there's lots of people who have it but don't have a positive test result for it. And then the infection rates. So testing has been better in other places, like China and South Korea, for example. South Korea has done an excellent job with testing. And so we can get more reliable data about the virus by looking at how it's going on in other countries where the data are more reliable than they are right now in the United States. Yeah, which is a great reminder that this is something that is global and it is not, it does not affect just one country at a time. And so we can learn about it by thinking about this on a global perspective. So again, this is just to reiterate what I just said, but labs that got false positives had to stop using this test, but it was the only test that was approved to be used. So even though some laboratories, for example, pretty notably, the virology lab at the University of Washington quickly developed a test, but there are differences in regulations about a test that you can use for research purposes versus a test that you can use for clinical purposes, because again, it makes a lot of sense that you don't just want a scientist coming up with something and then not running and not having oversight and then providing medical information based on those tests. So initially, even though there were other tests available in the United States, they couldn't be used to provide medical information. And so there are some certain emergency use authorizations that have been used. And so now certain laboratory developed tests, which again, all that means is the labs design different primers for qPCR reaction. Those are now becoming more widely available. And that's why you see at the beginning of March, for example, the University of Washington Virology Lab has started testing thousands of samples a day. So testing is starting to ramp up, but we are probably set behind by weeks because of this fiasco. And that has important implications for how treatment and how public health measures are being implemented. And I'll come back to this a little bit later. And so I have a question. So would you question the reliability of the primers? Yeah, it is possible that this was a primer issue. And I'm not going to dig too deeply into this, but for example, if you have bad primer design that allows them to bind less specifically than they should. So for example, if there's if a primer is 20 bases long, in theory, you should have a 20 out of 20 match. But if it's a 19 or 18 out of 20 match, sometimes it's good enough. And so then you can get false positives because it's matching up with something that's in your cells that shouldn't be matched by the primers. There's other things too, like for example, if the primers can fold and base pair to themselves, certain types of qPCR reactions are affected by that. And that's often a cause of false negatives as well. Okay, so now we know all about what's been going on with testing. So the next question is, can a test tell if a person has recovered from previous infection? So we know that the qPCR tests that are commonly used should be able to tell if you have virus RNA in a nose swab or anything. But what if you had it and your immune system fights it off and now you don't have it? a qPCR test would not be able to tell if you had it and recovered, but other tests that could be able to do that are currently in development. So they're not available yet. Again, we're really, really early in dealing with this. And so the pace of science has been really rapid, which is encouraging, but it still like takes time. So something to point out, and this is just basic, immunology, is that when we're infected, our bodies can produce antibodies that can bind to a virus and target for destruction. This is how we deal with most illnesses that we encounter, is that it takes a little while for your body to recognize it and make antibodies, but once it does that, it can effectively deal with it. And antibodies stick around, sometimes for reasonably short amount of times, but other times antibodies can stick around for very long time. For example, that's why if you have chickenpox once, it's very unlikely that you'll get it again, even 10, 20 years later. Whereas some other things, the amount of antibodies do drop off a little bit after a couple of years. The nice thing is that because antibodies stick around, if you can test for the presence of antibodies that detect the virus, you should be able to tell if the person has previously the virus. Also, this suggests that the person would be immune. And something I want to point out, this came out like 3 or 4 days ago. So when I talk about how rapid things are changing right now, a lab published a preprint. This is basically means that they posted some research that is going to be peer reviewed soon, but they're basically giving people, allowing people to look at it before it goes through peer review in an effort to make science, to be faster and transparent. My disclaimer for this is because it hasn't gone through peer review, you want to be careful. And so, for example, I've looked at this paper and been able to assess whether or not I think it's trustworthy, and I do. But it suggests that there are some research labs that have already had success in preliminary development of using assays that can detect the presence of certain antibodies. And it seems that when they're testing people, the blood samples of people who clearly had coronavirus earlier and have since recovered, that they can detect that they have antibodies. And this is really encouraging because, again, not only is it good, just testing in general is good, but you can also think, for example, if you can test if a person has been exposed before and then likely has immunity. For example, if you know that a nurse has had it and has immunity, that nurse might be the best person to go treat coronavirus patients now because they're not going to catch it again. And so it can make decisions that have really important public health implications. Likewise, if a person in your household has immunity, but someone else doesn't, then the person who has immunity might be the good person to go do the grocery shopping. Okay, so now we're getting into the more medical aspects of the disease, now that we've talked a bit about the virus itself. And so what is COVID-19 disease? I want to reiterate the terminology. So COVID is the disease, and it is caused by the novel coronavirus, which the technical name of which is SARS coronavirus 2, with SARS coronavirus 1 being the SARS outbreak that happened in the early 2000s. So this is the virus, and this is the disease it causes. The symptoms of the novel coronavirus is primarily a respiratory illness, and this is similar to SARS. The difference is that you tend to get a wide range of severity, ranging from some people who get it and don't even have symptoms to the point where they never even knew they were sick, to people who are ending up in the hospital with lung failure. So the main symptoms that have been reported are fever, a sort of dry cough, and shortness of breath, so trouble breathing. Most people, again, have a mild infection, ranging in severity from a cold to a flu, but a small but still significant subset of people do have severe illnesses, and that can lead to respiratory failure and death. And this is why there's a heavy need for ventilators in hospitals, because if people are unable to breathe properly, it's quite obvious how that can lead to death. And also, you may have heard a lot of information about people who are old are susceptible, but people who are young, it's the you're the ones who are safe. That's really a simplification, and I think it can be helpful to mention the difference in risk, but also a little bit dangerous if people take that too far. So I want to point out that, people in their teens and their 20s and their 30s, while they have a lower risk of getting a severe disease, there are still plenty of people who are in their teens, 20s, 30s who are ending up in the hospital in critical condition. And again, also, there's substantial evidence, especially coming from some of the studies coming out of South Korea, that even though younger people who are not immunocompromised have a lower risk of getting the severe illness, they're still perfectly able to transmit the illness to other people. And I think it's also really important to recognize that attitude of like, oh, only the sick and old are going to be victims, even if that were 100% true, which is not, is still like important. Like, these are people's lives. And the value of a life is not determined on whether or not that person is immunocompromised or under the age of 60. So yeah, take this seriously. How is coronavirus being treated? But right now, there's a question here. I heard in the news that Wuhan has created this vaccine. Does it actually work? I'm going to talk about vaccines in a minute. There are no vaccines available right now. Okay, so symptoms right now, the treatment is generally treating the symptoms because there are no known antivirals that are currently effective. My hope is that I think one of the best case scenarios is that we figure out that an existing medication that has another purpose ends up being effective at treating this, because that means that it has already gone through clinical trials and been shown to be effective, and then it would just need to be approved for a different use. But right now we don't have anything that's actually effective at like preventing it. Basically, the medicine is your immune system. And then basically the support that the medicine that we're giving people is just treating their symptoms so that their immune system has the best chance to fight off the illness. So people with mild symptoms generally are just stay home and rest, drink lots of water to keep hydrated, and the same thing you would do if you had a cold or flu. Also, it's very important that if you have symptoms of this, stay home, because if you are experiencing symptoms, and you're definitely at the infectious stage. And then people who experience serious illness, such as respiratory failure, this is why we're having so many people going to the hospital, is because, again, even though it's a low risk, if a lot of people get it, if it's a 1% thing, but 100,000 people have it, it's a lot of people ending up in the hospital. And we do not have, this is sort of the thing I'm going to highlight in the public health section, but we do not have enough ventilators to handle that. And so if a ventilator gives you a good chance of surviving, but every ventilator in the hospital is occupied, that makes the virus much more deadly, because if you can't get assisted breathing and you require assisted breathing to survive, that can lead to death, even if it was technically preventable. So yeah, ventilation and supplemental oxygen. You're basically supporting the body so the body can continue to do its job of fighting the virus with its immune system. So to get to the question that I just had about will there be a vaccine? So the answer is there will hopefully be a vaccine. There are no vaccines right now. And vaccines take, I'd say it's possible, especially given that this has become clearly a research priority worldwide of creating a vaccine toward this, that we might see something faster. But I'd say most reliable estimates that I've seen from experts in the field of virus development and just in general immunology, as well as clinical trials, suggests that we're looking at the order of maybe a year or more in terms of having both a vaccine that works, that has been tested, and is reliable. I'll get to the chloroquine stuff. I have a question in the chat, has been coming up very, very recently, so I'll get to that in a minute. And I want to point out that before I get to that, I'll mention the rest about vaccines, is that there are vaccines against other coronaviruses, but we use those vaccines on dogs often. for example. So this is some encouraging evidence to show that coronaviruses in general are able to be targeted through a vaccination strategy, which gives me, it's a little bit encouraging to know that a similar thing has been attempted and worked with a similar virus, which suggests that it may be a little bit easier to make a human novel coronavirus vaccine than some other types of viruses. And a question that I have in the chat is, do I like the idea of off-label use of medicines like chloroquine and zinc ions to inhibit some of the like proteins that are involved, such as the RNA replicase? And the question is the disease intestinal in paw friends like dogs? Yeah, so I'll answer the second question first. So the coronaviruses that infect dogs, there's also many that infect certain farm animals and stuff like that, are often associated with gastrointestinal diseases. So you'll see things like vomiting and diarrhea. So those are the things that dogs are often vaccinated against. Again, this likely has to do with the fact that although there's some evidence that, or there's in your GI tract, there are some cells that express the receptor for the novel coronavirus. Different coronaviruses have different receptors. So you can get different types of illnesses, especially the farther you go away in the family tree. And then to answer the first question about off-label uses to specifically target the things that we do know about the viral biology. So for example, what was mentioned in the chat was the RNA replicase, which is the protein that the RNA uses to replicate its genome. So I think that this is incredibly important. So right now we're going to be seeing a flurry of activity of people trying to see if they can use existing medicines and see if they're effective against the novel coronavirus. And what you're seeing here is also a hypothesis that because we know about some things about coronavirus biology, we can make a hypothesis that certain drugs might be effective instead of just having to do a shot in the dark. So I certainly think that one thing I do want to warn against is that there's some early suggestions that maybe chloroquine might be effective or something, but these are still so preliminary that I do not feel confident to comment on whether I personally think they will pan out or not. I just don't know. It's so early. And also I want to really caution you that we're hearing a lot of situations of overpromising in some situations by politicians. So I know that the president has been making some comments about chloroquine that I think are overstated, at least for now, about its effectiveness. And I want to caution people against buying into that too much, at least now. I really hope it works. But right now I have no evidence to suggest that or to make a good prediction about whether or not that's likely. Okay, and then I had somebody ask a question about, should I be wearing a face mask? And then questions, are there other zinc ionophores out there? Probably, I don't know. I would guess, but I'm not certain. And then, should I be wearing a face mask? So my quick answer is probably not. So face masks are good for a few things. They're good at stopping you from touching your own face, which we know can be helpful. And they're also good at stopping a lot of the spread of liquid droplets. So I mentioned that if you cough, you can spread liquid droplets like 6 feet. So if a sick person coughs and those droplets hit a face mask, that's going to reduce the amount of spread. However, improper mask usage, which is how most people wear masks, is reasonably ineffective. So I have this nice, I just thought this cartoon did a really good job at explaining this. So people who are at really high risk of contracting, like medical professionals, should absolutely be wearing masks. They may not be 100% effective, but they're going to reduce the possibility they get it. And also these people are often trained to wear them in a fitted way and to not sort of compromise how well the masks work. And if you're caring for somebody who's sick, for example, that's another situation where you know you are putting yourself right up against that, a possible case of transmission. So those would be reasons why a person should probably wear a mask. But to point out that if you're wearing a mask just as regular, just to try to prevent yourself from going to the grocery store and getting it, it's quite likely that the mask will be ineffective because there's a lot of people will touch the mask when they're taking it off and then they've contaminated their hands. Often you're getting other surfaces being infected. And so it's much less effective than actually making sure those masks are reserved for healthcare professionals. And also a really good reason to wear a mask is if you are sick, because again, then you can be containing, for example, the droplets that you're producing so that they're less likely to spread. And that's much more effective than if you compare these two ones, if using them as prevention from receiving it, as opposed to prevention from spreading it. So my short answer is, are masks effective? To some extent, yes, particularly if they're worn correctly and they're fitted, which is typically what happens with medical professionals. But there's shortages of them right now. And so the sort of benefit you could get from going out and wearing a mask is extremely low. And the danger of sort of taking those resources away from the healthcare professionals who are interacting, who are definitely having exposure constantly, is the bigger danger. Because again, you cannot be thinking about this outbreak in a sort of me versus everyone else sense, because the healthier everyone around you is, the less likely you are to get it. And so we need to be thinking about this on a broader scale. And again, this is a simplification, but I think that it was a really nice general demonstration. Okay. So question about how are people recovering from COVID-19? Are they immune? So I mentioned this a little bit before, but what we know from other similar viruses suggests that people should acquire immunity after infection. I have no reason to believe that as we start learning more about this, that we would see anything different, but we don't know for certain yet. This is my caution. But I want to point out that if we look at the related SARS virus, again, this is the SARS virus that caused the SARS outbreak in early 2000, that's very closely related. People who were infected 1 to 2 years later still had reasonably high antibody levels in their blood, which suggests that they're immune. You can't actually be certain that they're immune because we don't reinfect people with SARS to check. SARS is currently not in circulation. But this is just to say that we don't know for certain, but it probably is my answer for immunity. And we're really counting on that because immunity means that once somebody does catch it and recover it, then they're a really good person to be able to sort of go back into the workforce and also not necessarily be a vector for spread. Okay, and then I'm going to finish up by talking a bit about the public health aspects of this, which is, I think, what in the case of many people here is affecting their lives directly. So a lot of people are confused about why are schools and business closures, why are quarantines happening, why are other major changes happening? Like, why doesn't this happen every year for the flu? Why is this it seems like we've gone from 0 to 100 real fast. What's going on here? And so I'm sure that a lot of you have seen something like this going around, which is great. And I think that this is a really important way to be thinking about this. So I've mentioned that hospitalizations, when they do occur, generally require ventilators. And so if there are not enough ventilators, people will die, even if they might have been able to live if there were a ventilator available. And so if we can reduce transmission, that means less people get it all at once, which means that there are less people without hospital beds who need them. And so this is what people mean when they say flattening the curve, because this is like the curve. This is not actual data. This is a schematic that is basically demonstrating the general concept. And so here I want to really carefully explain this because I don't want people misreading this graph, because I think that it's very important, but I don't want you misreading it. So there was a group at the Imperial College London that did some mathematical modeling about, in mathematical modeling is a really helpful way to basically say, okay, we're going to make a prediction. It's going to be imperfect because we're making some assumptions and we can't measure every variable, but we're going to make a prediction about what's going to happen, and then see how that would change if we did different things. So when you're reading this graph, I don't want you to be treating it like a forecast. I want you to be looking at how the interventions change the general trend. So it's suggested by this modeling that if we just, no one did anything different, then we just kept living our lives, there was no school closures, nobody was social distancing, if everyone just ignored it, like lots of people would get sick really, really fast. And you'd see that very, very quickly we'd overwhelm hospital capacity, which means very quickly a ton of people are sick and not being able to get the care they need, which means lots of people are going to die in a way that would be, in a way for some of them that would be preventable if there were adequate hospitalization resources. So then what the researchers did was they plugged into their mathematical model differences that would happen if different forms of isolation, in their model, they suggested, okay, what would happen if we did 5 months of an intervention and then stopped? Again, this is why this is not a forecast, because they just picked one scenario, 5 months of intervention and then no intervention, which is very different than likely how it's going to be approached in reality. But it's helpful to show the main takeaway is that you can see with two different interventions, a moderate and a moderate, like we're going to close. If you have it, you should stay home. If your family member has it, you should stay home. Everyone stay 6 feet away from each other. You can see that this purple line, while the intervention's happening, the virus is spreading, but it's mostly staying under the capacity that the healthcare system can work with. But then if you just lift the interventions and stop doing them, then it bounces back. Because again, the virus is in the population, there's not immunity yet. So it's not like doing this for a little while just drives it to the ground and then it never comes back. They showed that a more severe intervention quelled it even more, and then again, it bounces back later with less people having immunity. So what your takeaway from this should not be that we are doing 5 months of, of isolation and then nothing. That was just part of the modeling. Your takeaway should be that these prolonged dramatic public changes, such as closing schools and really avoiding contact between people, flattens the curve. And so that's going to save lives. And the other important conclusion from that is that if we just stop, unless there is a vaccine available or unless there are drugs turn up that seem to be effective, if we stop this, then it's going to start picking back up again. And so I think that's a really important thing for people to recognize when trying to consider what is the next week, month, year going to look like. And yeah. So to sort of finish, and there's a question. So we need a year-to-year half so we don't hit surge cap. And then, so what does it mean in isolation? Yeah, let me go back to that to answer these questions. So isolation for an extended period of time. So you can consider like isolating being, if you have somebody who knows they have coronavirus, preventing them from interacting with like anybody other than like medical professionals and stuff would be isolation. Whereas quarantine would be like, for example, I'm doing something like a quarantine where I have no reason to think that I'm currently infected or anything, but I'm staying in my house anyway. And then, yeah, so these are different types of public health interventions that they were suggested. And the next question was, we need a year-to-year half so we don't hit surge cap. I'm not exactly sure what that question is getting at, but we definitely have to, yeah, we definitely are going to have surges if there are removal of the public health measures and that are likely to overwhelm healthcare capacity. And then how long will it take to get through this? My answer to this is I don't know. No one knows. We can make some predictions. I mean, if we look at, for example, some of the modeling that I just talked to you about, it's likely that we're going to be seeing things more on the order of months than weeks. But again, that's not a guarantee. That's just my prediction. But there's some encouraging things to consider. So if we can get rapid testing done a while, I want to point out the difference in these graphs showing, you can see that the United States, there's some incomplete data here, but at least early on, has had very delayed testing, which we already know about. Italy is sort of ahead of us in this, and in a somewhat similar situation where they currently are having an active outbreak. But South Korea has handled this very differently. So South Korea has been doing very, very aggressive testing. And so by this, I mean, for example, testing people who are asymptomatic, testing people who, like not just testing the most severe cases. And in this way, they've been able to identify a lot of people who are infected but didn't know, and then able to implement, isolate those people and say, like, you are infected, do not contact anyone else, like, don't go to the grocery store, don't do anything. And so by doing a lot of testing, you can actually almost be a bit smarter about how this is implemented. And because, for example, I'm at home and I don't know if I have coronavirus. I hope I don't. But again, like we said, like, I have no reason to expect I do, other than the fact that if I do, it's asymptomatic. But I don't know. I haven't been tested. Neither have any of my housemates. And so, like, eventually one of us will have to go to the grocery store again. And so if we aren't tested, we don't know who, if any, if all of us are infected. And so you have to just sort of operate under the assumption that everyone is infectious. If you don't know, you kind of have to assume everyone is. But with aggressive testing, you can be a bit more strategic about it. And so I think that as testing ramps up in the United States, I hope that it becomes very widespread, because that is really important for informing public health decisions. And we can see that that has actually helped South Korea, and in addition to other very strict containment measures, help them contain the virus much better than some other countries have been able to. So that is what I have for my prepared presentation. I want to also leave you with my sort of plea that this presentation has been based off, I spent a lot of this week reading lots of primary research, reading reviews on coronaviruses, looking at the research that has been done, the new research that's been coming out. Part of this is because I'm trained as a biologist, and this is part of my job, is to be able to read and understand research. I also understand that for a lot of people, this is not accessible because it's written for experts, and so it's written in a language that people can't do. So please look for trustworthy sources. I've been sending everyone who asks me to the CDC website, because they have really comprehensive and information that they're keeping up to date as much as they can. That's pretty reliable. Also, if you're looking for journalistic resources, I found that when I've been going through and comparing what I've been reading like on news sites to what I've read by actually looking at the research myself, I found things like the New York Times, Wall Street Journal, and The Atlantic have been really excellent in reporting accurately and also in a easy to understand language. And then if you see something that's not verified, even if you think it might be helpful, don't share it unless you're sure it's true, because a lot of people have been very well-meaning, but are spreading misinformation, and this is causing confusion, which actually makes it harder for people to make educated decisions. So so I see some things about like PubMed. So like, when I'm looking for research, I'm going to PubMed and Google Scholar, in browsers. And a lot of journals that are still behind paywalls are lifting the paywall restrictions. So you don't also, if you are like, for example, teachers, if you're trying to get something that's behind a paywall, you can email me and I'll send you the PDF. So please don't pay for research articles. Okay, and so I have some questions now. What is the takeaway of the presentation? In other words, what should we be should do for the most likely extended period of time? So again, this is just my perspective of this. In many ways, I don't know the right way to do things and the wrong way to do things, but I can tell you that what I've been, telling all the people I know and love is that you should be understanding that this is a very serious situation and that the public health measures that are being put into place are very warranted. So I would recommend, as much as you are able to, like make decisions that are reducing transmission. So if you have to go to the grocery store, like you don't have to like stay in your home and like, boil your shoes because you ran out of groceries. Like you should, you can still go to the grocery store, but you should be strategic. Try to see if you can plan ahead so that you only have to go to the grocery store once in the next 2 weeks instead of twice, because that's going to reduce transmission. You should not be meeting with people unnecessarily. So for example, I have regular contact with my roommates because I live with them. But I have lots of, if I go and go hang out with a group of friends and say, oh, I'm only meeting 4 people. Okay, well now those 4 people have contacted me and then I've contacted all my roommates. And then if they go and meet up with 4 friends, it's very quickly to make a big chain connecting a lot of people. And so the more you can cut off the chain of people you contact and then people you go on to contact, the better. And again, this, some people will make decisions to, go visit their parents and, that is fine. But you should be, anytime you're making the decision to encounter another person, you should be taking that very seriously because you should be operating under the assumption that it is completely possible that you are infected asymptomatically. This also means that you should just be operating with extreme caution. I also want to say that this does not mean that you should panic. When I say that this situation is serious, that does not mean you should panic. There's a very different, those are sort of the two ends that I've been seeing, is people who think it's not a big deal and people who think that they need to panic. We need to have something in the middle of there, where you're taking it seriously and still, and thinking calmly and rationally. But yeah. People have other questions, feel free to throw them in the chat box. Happy to stick around and answer them. Okay, so I'm being asked, can you give me the citation for the sequence data? What is the site used for amplification? Let me see if I can find so you can figure out the exact primer sequence by going to the CDC website. So that's for the CDC test, but again, that's for the CDC developed test. There are other tests that are gonna have different sequences. And so again, I don't personally, off the top of my head, know which sequence that the CDC ones are targeting. I'm pretty sure that that if you go to they have like a protocol that you can download on their qPCR protocol, and it has the primer sequences listed inside of it. Let me see. I can. Can you still see my screen? Oh, I'll share it in a second. Um, okay, so I pulled up the CDC protocol and it should have the primer sequences in here somewhere. Oh yeah, here's a link to some of the, okay, it took me back to the same website. Yeah, you should, somewhere in here, they have the primer sequences. Might be an appendix at the end. You can also, I definitely know that the University of Washington virology site publishes their primers. Yes, so here's the package contents. Yeah, I'd have to sit down and yeah, got me to the site. There's also the UW virology website that they have developed a lab-based diagnostic test that uses different primers, and you can actually like just go to their website and they have their primer sequences listed. Okay, see a question here. Do you think climate change has an effect on the disease? Will we see it soon? And will climate change make epidemics and pandemic more prevalent or normal? That is a really great question. Also, I'm going to go back to, I don't know where my, here we go. I'm just going back to my, so I expect that climate change is certainly having effects, but not necessarily in the most direct way you might think. So for example, climate change is likely to lead to things like climate refugees, who are people who are displaced by climate change. And this can affect, for example, the spread of disease by putting people in close quarters, or making sure, or basically meaning that people are not having access to adequate resources. I don't think that if you're thinking about climate change in the terms of like, if the weather gets hotter, does that change the spread of the virus? There's no good reason to suggest that there will be any substantial weather-based change to how the virus is spread. We do know that in general, often you see lower rates of spread of many diseases in the summer for various reasons, including, but not limited to, people spending more time like doing outside work and therefore not being in, inside. It's not that being inside is more dangerous, it's that people are being inside in public spaces more often than being outside in public spaces. So more close contact with people. Also, there tends to be differences in humidity, which can affect, for example, the efficiency of aerosol and droplet-based transmission. But from all accounts that at least I've been able to see so far, weather-based, and again, this is weather is in general, like going to the summer, it'd be great if that gives us a little bit of protection, but I think that if there's any effect on the weather, it's not going to be very substantial in comparison to the size of the outbreak otherwise. But yeah, climate change is something that I think is affecting the way that we live our life in like every aspect. And I think that there are some important parallels between climate change as being a really long-term thing facing our society that's going to require collective action to combat, in the same way that this is a very short-term challenge to human society that crosses borders and is going to require very short-term collective efforts like staying home, closing schools, and whatnot to combat. But yeah, so they definitely are related, and often in ways that are complex. Yeah. Okay, the question is, do you have any feel for the ACE2 receptors upregulation in response to high blood pressure medications, those patients that are more at risk? I'm going to be very cautious when I answer this question because it will involve a lot of speculation. So the ACE2 receptor is on many cells, but most notably it's on type 2 lung alveolar epithelial cells. So these are cells that are in your air sacs in your lungs that are involved in gas exchange. So the reason you breathe is because you bring air in very close contact with your blood circulation, and that allows for diffusion to remove carbon dioxide and bring oxygen into your circulation. And so those are the cells that have the ACE2 cells. So those are the cells that are targeted by the coronavirus. And the ACE2 receptor can be involved in regulation of high blood pressure because it's involved in a system called the angiotensin-renin system, because again, it's called the angiotensin converting enzyme. And so I'm not going to go into the angiotensin-renin system right now. Anyway, so my speculation is that if the ACE2 receptors are upregulated, that may make it easier for someone to get the infection because there's more docking points for the virus. So you'd say like the probability of a virus being able to enter a cell would likely be higher. Again, I'm speculating on this. I don't know for certain. So there might be good reason to think they're more at risk. It also, I think that I've been seeing a lot of people conflating angiotensin converting enzyme with angiotensin converting enzyme 2, which for the people who are interested in engaging with us on this level, it's important because they have opposite effects on blood pressure regulation. Because, yeah. So definitely differences in ACE2 expression, I would speculate would likely affect the patient's risk. And I'm certain there are people studying this right now who know much more about it than I do. Okay, I've got 2 questions here. First one is the news that home testing COVID-19 is coming up, is that reliable? I would say like sort of yes and sort of no. I think that a lot of it depends on the time scale that you're talking about. I'd expect that like I don't think there's going to be like a pregnancy test type test where you can just like do it yourself, at least in the near future. Will that change? I hope so. But that would involve substantial, like that's going to be much more different than designing a new qPCR primer and then just doing the same qPCR reaction with a different primer, which is how a lot of the new tests are being developed. So I don't know of a home test that is currently available or that will be available very soon. And I don't know what time frame you could expect, if any, them to appear. Coming at March 23rd. I'm not sure what source you're referencing, and so I can't comment on whether or not it's reliable. You know, I'd be really happy if it was something that was true that I didn't know about, but I don't know about the test you're referring to. Oh, somebody said something like holding your breath for 10 seconds. Does that really work? No. So there's a really common thing of, it's a whole string of facts about the coronavirus that have been spread widely on social media and misattributed to a Stanford doctor. So some of the things have included that drinking water washes the virus into your stomach, so it's killed by the stomach acid. You should absolutely be drinking water, but that's not how that works. There are people who have been saying that, you know, drinking cold water versus hot water, which has no basis in science. And there have been people saying that to tell if you have it, hold your breath for 10 seconds. And if you can't, then you have like damaged lungs. And that is also very incorrect and dangerous also because if people are treating that as medical advice and therefore trying to make self-diagnoses based on something that is not actually diagnostic of whether or not you have this, So for a good example of why this is not effective is that, one, we know that people can be asymptomatic. So there are people who have no symptoms. So if they held their breath for 10 seconds and they would be fine. There are some people who have other underlying conditions affecting their breathing. So somebody might have asthma or some other pre-existing condition that makes them unable to do that without coughing in some situations. That does not mean that they have COVID-19. So that's an example of a test that will give you false negatives and false positives, which you now know what that means. And so it is not a reliable test, and it does not have, if you hold your breath for 10 seconds and you cough, then there's lots of reasons that could be true. And not all of them are COVID-19. So and I want to point out that I've seen this shared, at least on my own Facebook feed, like 20 times. And it's people who are nervous and they're scared and they see this and they say, this looks like a scientist wrote it. But it's like literally like a screenshot of someone's notes app. But they look at it and they say, oh my gosh, I'm confused and I'm scared, and here's some information that makes me feel more comforted because knowledge is power, and I'm going to share it. But the problem is that people are doing this with correct information and incorrect information, and then that becomes problematic. So I'm really glad that you brought that up, because I want to dispel that as being a myth, and make sure that my general advice is, if you see something on Facebook and you can't confirm it, just like, that doesn't mean it's false, but it doesn't mean it's true. So just pretend you didn't see it. Don't share it. Or you can, if you saw it and you're curious, then go and look and see if you can find a reliable source that backs it up, and trust that one instead of the, so like if it's on a meme, maybe it's true, maybe it's not, but like, don't get your medical information from memes, because I've been seeing a lot of that on my Facebook feed. Memes are great. They're really helpful for like connecting socially to one another and spreading culture and, helping us make sense of the scary situation, but they're not the ideal form of spreading medical information. Okay, and so I have a question from Mrs. Famoso asking if I can talk about how I got to where I was, my educational path, what I'm planning to do for after my PhD and advice to students. So it started, I was a student at Colonie. I really, really when I took ninth grade biology with Mr. Goldberg, I think that I'm pretty lucky, but I had sort of a light bulb moment that I don't think a lot of people have, where I was just like, this is what I want to do with my life. I just knew right away. I was like, I really like learning about biology and talking about biology. So I took all of the AP Bio, Molecular Bio, an independent study in biology when I was in high school. And I wanted to get research experience. So when I went to college, I chose a college that had a lot of research opportunities for undergraduates. So I went to the University of North Carolina at Chapel Hill, which is a research university in central North Carolina. And so I studied general biology there. And I worked in a laboratory there where we studied a few different things, but they sort of fall under the category about how do you build proper skin, and how do you build proper structures in your mouth. So what I was studying a lot is, how do cells decide if they're going to, so if you're making skin, you need to make your skin have more surface area, and you need to make your skin thicker. And so cells have to actually divide in particular ways to make sure that you're balancing those decisions. So I studied that, and also worked on how the molecules that hold your cells together, so adhesion molecules, there's a reason why we're not puddles on the ground of cells. We have some cells that stick to ones and other cells that don't stick to each other. And so I was studying how those are important for making a palate. So the roof of your mouth fuses, and we know that if that goes wrong, you can get cleft palate. I also did research at during the summers at UAlbany. studying salivary gland development in mice, and at Columbia University studying stem cells in fruit fly intestines. So I got a lot of opportunities to sample things that I really liked. And then I'm doing my PhD now at the University of California at Berkeley. And so what a PhD is essentially, it's a degree where, the way I think about it, it's like you're doing research and a little bit of classwork with the goal of becoming an expert in a particular subject area. So the subject area that I currently study is broadly developmental biology, which I define as the biology of how do you build a body. And the particular sort of the tiny little corner of that I study is that I study how the cell adhesion molecules, which I studied in undergrad, so I love them, how those cells or those molecules are involved in cells making decisions like whether or not to grow, or how those molecules, for example, are involved in organs growing to be the right shape and size and then stopping when they reach the right shape and size. That's my very general pitch about the research that I do. But most of my day-to-day life is doing, well, I guess not right now, but before this is doing experiments where I use fruit flies. I study how they develop their wings. And I do things like genetic crosses, and I use a lot of microscopy to try to sort of answer little tiny questions that by answering each one of those, I can understand more broadly these big things. But I also do some teaching. So I, like 2 semesters ago, I was a teaching assistant in a human physiology course, which is now becoming very useful because now understanding human physiology is really important for understanding this disease. And right now, like actively during this pandemic, I am TAing a developmental biology course. So I've got a few more years before I can get my PhD, so I'm sort of in the thick of it. They usually take like 5 or 6 years. It's much more like having an intro level job than it is like being in school the way that you know it. Like I get paid, like I mostly don't take classes. So it's very different than like high school or college. And then afterward, I'm most interested in doing university teaching. So I really enjoy talking about science and helping other people understand science. But I'm not exactly sure 100% what I want to do, but that's fine because I got a few years to figure that out. Thank you all in the chat. And if people have any more questions, let me know. I'm happy to stick around.
Danielle, this was really wonderful. Really wonderful. And I can't thank you enough for doing this for us and for the community here in South Colonie. Yeah. And my heart has swelled 1,000 times watching you speak. The level of how proud I am of you. And I know Mr. DeMarco is probably saying the same thing. And when Mr. Goldberg sees this, he's going to be saying the same thing too. We knew you when, and we have now the recording to prove it.
Yes. Yeah, it's nice to be able to help out my community at home. It's, I'm like 3,000 miles away, so it's a little bit hard, but this is one of the advantages of technology. And also it's nice that a lot of the times, my day-to-day research, I'm working on something that's very, very specific, which I find very interesting, but it does not have obvious medical applications immediately or anything. Like it's not going to immediately change anyone's life. It will help us know a little bit more about the big questions of the universe, but it's nice being able to use my expertise in a way that is directly helping people in the short term, which is a little bit different than how I'm often using it.
Absolutely. Well, again, I really appreciate it. Yes. I echo Mr.
Yeah, thank you for
DeMarco's.
taking this. Oh boy, yeah. Well, now we'll see. Like,
Yeah, we'll see now.
maybe everyone who did their PhD in 2020 is going to have much longer. My program tends to do 5.5 to 6 years. I'm currently in my 3rd year. And yeah, I've got a lot of research to do before I'm ready to graduate, but most of that usually happens in the 3rd, 4th, and 5th year. So,
well, we're going to hope that we're on a shorter time scale than what you presented today. But
yes.
no matter what, we know, yeah, you're gonna be one of those rockets. You're gonna be one of those stars that we point to. You're gonna be on our wall of fame one of these years. I know it. Back at the high school, and I'll make sure that I send you the link to this so that your mom can see it for sure.
Yeah, absolutely. Thank you.
Thank you, Danielle.
Yeah, thank you so much for organizing this.
Oh, you're welcome. You're welcome. I'm going to stop the recording here.
Okay.